downstream of the receptor. So there's all these different levels of actual control that we're trying to figure out. And then with respect to things like other kinds of signaling that, and this has more to do with, let's see, this one here would be, for I know it's supposed to be only talking about triple negatives, but this would have relevance also to even ER positive, ER-PR positive tumors. I believe PI3 kinase signaling inhibitors are now being considered strongly for triple negative and for ERBB2, aren't they? Anyways, I think about that. But I mean, this is more of a different subclass of breast tumors. And the point is, the stiffness plays out here. So in a stiff environment, in response to growth factors, look at the magnitude of the signal as compared to a soft matrix. And just back in our animal model, FAC activation was reduced, as I told you, but signaling through this part was reduced significantly. And that's one of the key things that's thought to be involved in driving an invasiveness of tumors. So in terms of richness of where to explore, what kind of things a person could think about, I just put this up here for those, any of the postdocs that are actually doing any of this kind of signaling work, that this is a beautiful review by Moranti and Brugge in Science several years ago that looked at all the different means and by which integrins can modify growth factor receptor signaling, and to begin to think about whether or not force and how force can play a role in each and every one of these different components or ways of transactivation of the receptor. I showed you that with the PGF. Receptor modification or modulation, which I showed you with the ERK signaling. Receptor compartmentalization is something that we're now actively pursuing, or coordination of pathways. And we have some work also going on gene expression relation. I thought it would be prudent, and I just mentioned some work that was published a number of years ago by Heinz's group that actually thought about TGF-beta activation. So what they showed, and this is a paper to go back and look at, is that you can change the activation of growth factors even in the extracellular matrix. So you need not even think about how the cells are responding. So one of the really key regulators is TGF-beta. That's been implicated so many times in terms of tumor progression and tumor therapy and what have you. And so there's some really nice work that showed that the ability of TGF-beta to be activated in the stroma of a tissue, in this case was just in a culture, but they were showing that the stiffness of the extracellular matrix enhances the efficacy of activating TGF-beta. So, that's a completely indirect way that the stiffness and cross-linking of the matrix can influence how that tumor behaves, because you could actually activate more TGF-beta. So, and basically in this particular paper, they talked about, compliant matrix through integrins. We're not getting a huge amount of integrin, oligomerization, and there's not a lot of cell contraction that's happening. And so you've got your TGF-beta in association with its latent binding protein. And so they're all kind of sitting there, and it's like tugging at it, but it's not really doing a really, really good job because the cell is not able to really exert a lot of force on the matrix. You stiffen that matrix, then the cell's ability to actually pull on and allow for the activation of TGF-beta is enhanced a huge amount. So that's like yet another example of how we can imagine that stiffness of the matrix can actually influence tumor behavior. So I put this up here just as a reminder about forces in tumors, and what I've been talking about here is the stiffness of the matrix and how that can influence how the cells are behaving with respect to the growth factors. But what I'd like to just give you one little tiny vignette about how the compression forces can also play another indirect role in terms of how tumor evolution can occur. So this is a paper that was published, it's actually a really cute paper. It was published a number of years ago about mechanotransduction by modifying basically the actual free space of the growth factors that are actually out there. So, you have a tissue, and so what they were showing is if you look at, and this is done in the lungs, but it's equally relevant for cancers, for anything where you're actually changing the mass of the cells and modifying that in the tissue space. So what they were showing is that you can have a little lung alveolus, and they noticed that in certain when they actually had a forced sort of ventilator where they're kind of putting a compression load on these alveolar cells, that they noticed, my gosh, there's all this activation of this growth factor. In this case, is EGF receptor. They're going, well, how do you get that happening? You know, are you actually releasing growth factor, or what are you really doing? So they did a bunch of cute studies that I don't have time to go into all kinds of belabored explanations for, but essentially what they did was they could demonstrate that what you were really doing is when you're actually applying a compression load, what you're really doing is you're actually reducing the free space around the cells. And so the cells are producing these growth factors, and what you're actually doing is you're increasing the apparent ligand concentration because you're reducing the free space. You're looking at it because you've read the paper, right? Did you like the paper?
That's very good.
Yeah, okay, so am I doing reasonable? Yeah, okay. So essentially what you're doing is you're reducing, you're pushing out, your sort of compression load, you're pushing out, and any free space is being reduced. So if there's a growth factor out there, the concentration of that growth factor is actually increasing relatively because the free space is decreasing. You're not changing the growth factor. You're not doing anything. You're not increasing its shedding. All you're doing is causing a physical change in the environment around the cells. And so I think they use these little transwells, and they could actually apply a force to these transwells, and they could show, if you had a pressure gradient on versus not. And the main thing is to look down here is that you were actually getting this increase in the phospho-EGF. So you were actually increasing the receptor activation without changing anything, without changing the amount of receptor or of growth factor that was expressed or released, simply by shrinking the free space and changing the apparent concentration. So those are all things to think about on that level, what happens. Well, what about the cells? You've heard over and over, your Weinberg and Hanahan, this is it, we know all the explanation, this oncogene does that, this tumor suppressor does that, beautiful, elegant work. We're not going to throw all that out because that happens to be really important. So what about intrinsic factors? Could it be that they're actually modifying mechanotransduction? Are they actually working in hand, or are they completely separate? So, that's kind of an important question, and I think it has barely been scratched with respect to looking at it. But the idea, of course, for most people is you got a mutation, various different, tumor suppressors kicked off and oncogenes enhanced, and then eventually you make an invasive cancer. And there are people like John Massaquet that's running around saying there's this particular mutation that drives this metastases and not. How do we understand that? Again, is it alphabet soup, or can we simplify it down to some kind of principles? And so basically, one of the things that we do know is that that metastatic potential of cells. There's certainly quite a bit of evidence that cells that become metastatic change their viscoelasticity. Again, there's that crazy thing, the viscoelasticity. They change their physical materials properties. So what drives that, and is that really relevant? So basically, we did some experiments a long time ago that showed that, of course, we and others could show that normal cells, we put them in a soft gel, make these little organotypic cultures. Tumors make aberrant little structures. These guys make tiny little nascent adhesions, as I showed you, because they're responding properly to the stiffness, right? If it's really soft, they don't make big adhesions. If it's really stiff, they make big adhesions. The tumors are confused. So they make these large focal adhesions even though they're in a soft matrix. And it turns around, if you start looking at their behavior in a soft versus stiff, while the normal guys tune their stiffness to the matrix, to their environment, the tumor cells just don't care. Now, what we know now is we probably modify that. They're just kind of, they're cells on speed. So it turns out that they're really hyper-responsive to mechanical cues. And so one of the things is that we know that they have elevated forces. And it turns out that for tumors, they just nucleate and they exert a ton of force. So the tumors are really driving these and nucleating these focal adhesions like mad, and they're really cross-talking like crazy to these growth factor receptors. In this case, I'm showing you EGF, but there's other things that are also implicated. So it has relevance because you can start to look at different kinds of oncogenes, and several different oncogenes we think, whether they're ERB2 or RAC or RAS or EGF, it turns out that almost all of them seem to be changing how the cells respond to mechanical cues. In other words, they're changing the mechanical behavior of the cell. And that actually is relevant because then we can start imagining, are tumors tension addicted? So here's an idea that if you have a tumor cell that's exerting a lot of force, well, you can inhibit those forces. You don't even have to go into all the regular signaling things. You don't have to use Herceptin or any of these other kinds of drugs that are targeting these signaling pathways that we're thinking about clinically.